helicase. This is the unzipping enzyme. If you recall that DNA has two strands, you can think of helicase as unzipping the two strands of DNA. Helicase doesn't have a hard time doing that. DNA polymerase, the builder. This enzyme replicates DNA molecules to actually build a new strand of DNA. Primase, the initializer. With as great as DNA polymerase is, poor DNA polymerase can't figure out where to get started without something called a primer. Primase makes the primer so that DNA polymerase can figure out where to go to start to work. You know what's kind of interesting about the primer that it makes? It's actually a piece of RNA. Ligase, the gluer. It helps glue DNA fragments together. More about why you would need that a little later. DNA replication starts at a certain part called the origin. Usually this part is identified by certain DNA sequences. There can be multiple origins within the DNA. At the origin, helicase, the unzipping enzyme, comes in and unzips the DNA. SSB proteins, that stands for single-stranded binding proteins, bind to the DNA strands and they keep them separated. Primase comes in and makes RNA primers on both strands. Now remember, that's really important because otherwise when DNA polymerase comes in, it wouldn't know where to start. Now DNA polymerase can get to work. Remember, it's the important enzyme that adds DNA bases. But the other strand will make it a little tricky. DNA polymerase can only go in the 5' to 3' direction. So RNA primase, it has to set a lot of extra primers down to do that, as shown here. It takes longer too. This strand is called the lagging strand, which is pretty fitting. On the lagging strand, you tend to get these little fragments of synthesized DNA. They're called Okazaki fragments. Amazing name. The primers have to get replaced with DNA bases since the primers were made of RNA. Ligase, remember that's the gluing enzyme as I like to nickname it, has to take care of the gaps in the Okazaki fragments. Now at the end, you have two identical double helix DNA molecules from your one original double helix DNA molecule. We call this semi-conservative replication because the two copies each contain one old original strand and one newly made one.